Hey guys, in previous video we have talked about the molecular biochemistry basics, in which uh, we talked about the basic structure of the chromatin and how the DNA is formed by these nucleotides. So learn about the structure of nucleoside and nucleotide. We discussed that nucleoside is made up of base and deoxyribose sugar. So just sugar and base is going to form the nucleoside, plus if we are gonna add the phosphate part, it will form the nucleotide. And we were discussing first about the base. So in base we have learned it is of two types, purines and pyrimidines. Purines has two rings, and adenine and guanine are the two components, and pyrimidines have one ring, and three pyrimidines are cytosine, uracil and thiamine. We discuss its relationship that cytosine deamination will form uracil, 5-methyl cytosine deamination will form thymine. So indirectly we form the relationship that if we do the methylation of uracil, it will form the thiamine. Thiamine is found in the DNA and uracil is found in the RNA. We don't have to particularly remember about each purine and pyrimidine structures, but we have to remember the components that are used in the formation. So we remember that the first nitrogen is going to be formed by the aspartate, then second and 8 carbon is by tetrahydrofolate, then third and 9 nitrogen is by glutamine, and 6 carbon is by carbon dioxide, and remaining, that means 4 and 5 carbon plus 7 nitrogen will be formed by glycine. So you can remember that in the first, third, 7 and 9, this 4 position will be occupied by nitrogen only. So it is an odd number except the fifth one. So 1, 3, 7 and 9. 1 is by aspartate, 3 and 9 is by glutamine, and 7 nitrogen is by glycine. All other will be formed by carbon only. So 2 and 8 is by tetrahydrofolate. We remember that it has tetra means 4. So 2 and 8. And the 6th carbon is by carbon dioxide. And the 4th and 5th carbon is by glycine. And it is a structure of glutamine. So in 6th carbon position, it is gonna have this carbon dioxide. Sorry, not carbon dioxide, it is given by the carbon dioxide. Now the structure of the pyrimidine, we learn that it has mainly two components, aspartate and carbamyl phosphate. So carbamyl phosphate, we will learn that it is formed by again two components, glutamine and carbon dioxide. So it is gonna form the second and third position molecules. So carbon is at second position, nitrogen is at third. So here we have to remember only first and third is going to be occupied by nitrogen, not fifth. All other are just carbon only. So second carbon is by carbon dioxide and nitrogen is by aspartate. So combining it, we will form the carbamyl phosphate, and all other are by aspartate. This thing is little tricky to remember the structure and the components that are forming it. You have to revise it on the last day to remember, I think. Now the synthesis part. So synthesis can be done by two types, either de novo synthesis or salvage pathway. What is de novo synthesis? De novo means you are going to synthesize purine and pyrimidines from its raw components, that is as we have discussed, the amino acid, carbon dioxide and the tetrahydrofolate are taking part to synthesize this purine and pyrimidine. So you are going to synthesize all this from this raw materials, while salvage pathway is it is only for the purines. So salvage pathway means by the catabolism of nucleic acid we are gonna get this free bases, and we are again going to use this free bases to use the new nucleic acid. Okay, let's see. So now first let's discuss about the de novo synthesis pathway. First about the purine. So purine de novo synthesis is very lengthy and very hard to remember. That's why they haven't given the particular steps that is there. I mean, there is actually 10 enzymes are used after this, but you don't have to remember any of them. It will be very confusing. As you can see here, these are all the steps. It's just for reference. You don't have to remember any of this. So now let's see. So first, our sugar part, ribose 5-phosphate. We know, so briefly discussing the sugar part in the nucleoside, we know there is an HMP shunt pathway, that is a diversion from the glycolysis pathway. So this HMP shunt pathway will use this glucose 6-phosphate dehydrogenase enzyme and will form after some steps this ribose 5-phosphate sugar, and this sugar are we going to utilize for the formation of nucleic acid. So this sugar we have ready, but now we have to synthesize the base from this. So as we can see, the sugar we have is ribose 5-phosphate, and from this we are going